All right, everyone. Welcome to our discussion on Chapter 8. We're now going to move into the nervous system. And before we go too much into the nervous system, let's kind of put down a flow chart, nervous system flow chart. Nervous system. And first, the nervous system is going to be subdivided into two sections. One is going to be called the central nervous system. And the central nervous system consists of the brain and the spinal cord. And on the other side, we have the peripheral nervous system. And the peripheral nervous system is everything outside the brain and spinal cord. And the peripheral system is further subdivided into the somatic and the autonomic. Somatic is conscious muscle control, as in you have a pen in your hands, you're taking notes, you choose to do that, that is because of your own free will. So somatic is skeletal muscle movement. And on the other hand, we have autonomic, meaning automatically, of course. And this is further subdivided into two systems. One is called the sympathetic, and the other is called the parasympathetic. We'll be going into them ad nauseum later on. Sympathetic and the parasympathetic. You'll be going into those later on. Let's define some other terminology since we're here. So we're going to use two terms when we describe neurons. Well, more than that, but these are just two of them. Afferent means delivers sensory information to brain. And on rare occasions, well, not rare, depending on how your day's going, could also be the spinal cord. So what this does is everything that you interpret in the world is going to be fed to your brain. Your brain is going to process it. For example, hearing, smell, taste, touch, how we interact with the world is going to be received by our brain and processed. Now the other is going to be something called an efferent neuron. E means exit, so efferent neuron is going to be motor neurons that respond, I think that's a good way to put it, respond to stimuli. So this means that you are going to have a change in your muscular system. To give you an example, your eyes are currently interpreting visual signals from your computer and auditory signals from, well, the computer audio as you're learning about this amazing lecture. So your response is going to be your efferent neurons are going to write down notes. I hope your efferent neurons are writing down notes. I hope you are not using them to play an online game, something like that. That's the difference between afferent and efferent. Once again, somatic system is conscious muscle control, where autonomic system is automatic, as in it is self-regulatory. We have no conscious control over it. If you think about it, we don't have conscious control over much in our bodies. Mainly just our brain. Skeletal muscles only move because our brain politely asks them to. You'll see about that later on. The reflex arc for the nervous system is pretty straightforward, thankfully. You are going to have an afferent neuron. And let's just go ahead and draw a neuron just so you kind of get an idea of what it looks like. So neurons usually have something that's going to receive information. It's called a dendrite. And they can have multiple dendrites. In fact, neurons in your central nervous system can have over, I think, thousands of dendrites. Now we see this central part here is actually called a cell body. It contains a nucleus. This is the living part of the neuron. You're going to see a long sort of a tube. Call this tube the axon. Then you're going to see the axon terminal. The axon terminal is the site where neurotransmitters are released. So once again, dendrites, cell body, the living part of the cell. This long tube is called the axon. These points at the end, they're called the axon terminal. All right, so this is going to be an example of an afferent neuron because it is going to be a sensory neuron. Anything that interprets sensations is an afferent neuron. Then it is going to deliver the information to the brain. Let's draw something that looks like this. Nothing fancy, just gives you an idea. So this is the brain, and the brain is the major integrating center. Because it is going to receive signals and decide what the outcome should be. Then, of course, on the other side, we have, not going to draw this neuron really as fancy, but we then have the motor neuron or efferent neuron because it's going to innervate some sort of muscle, tell it how to change its behavior. It's going to be the efferent neuron. Think of E for exits. Now, on occasion, the integrating center can be the spinal cord. On occasion, it can be the spinal cord. This only happens during something called a reflex. So sometimes you'll find yourself in a situation where you need to react so fast because if you don't, then you will incur bodily injury. Famous example is putting your hand on the burning hot stove. So if those receptors in your hand, once they detect that something's really wrong, if they feed it to your brain and your brain has to process it, by the time it formulates a response, you could already have major burns. So what happens is that on occasion, a reflex will be interpreted by the spinal cord, and it will give a twitch response, a very fast response to protect you from injury. So it still does use an afferent neuron and an efferent neuron, but instead of the integrating center being the brain, the integrating center is the spinal cord. That happens during a reflex. All right, so now we're going to talk about something called axonal transport. Exactly how does this thing work? Because a neuron is a cell, it just is a cell that's different than what you are used to. So we're going to talk about now types of axonal transport. And before I do that, I am going to draw a neuron again. So again, we have the dendrites, they receive signals. Draw a couple dendrites here. Drawing dendrites is kind of therapeutic. Really kind of a really great way to unwind from a long day. Just go ahead and draw some dendrites. It helps. I know you guys are thinking like, I think this professor needs to get out more, and you know you could be right. But in the meantime, this drawing dendrites kind of gives me a sense of calm. So anyways, I'm going to draw here the cell body. This is the nucleus. It is a cell, and it needs to synthesize neurotransmitters. It's going to have a rough ER, just like many other cells. It's also going to have a Golgi. Go ahead and draw the Golgi here. And it's also going to have other things like the powerhouse of the cell, the mitochondria. Did not learn how to do taxes in school, but I did learn the mitochondria is the powerhouse of the cell. I'm sure you all have seen that meme. And at the end here, we're going to have the terminal axon. So what's going to happen is that we are constantly transporting materials from the cell body to the axon terminal and vice versa. Now, the first kind of transport is going to be called fast axonal transport. So we'll go ahead and write that here. Fast axonal transport. And this moves things that are very small, like neurotransmitters. So mainly neurotransmitters are moved through fast axonal transport, or things that synthesize neurotransmitters. And it moves pretty fast, about 400 millimeters per day. That's a pretty good pace. Okay, and fast axonal transport goes from the cell body to the axon terminals. Now the next type of transport, which I'll draw here in sky blue, will be slow axonal transport. And this is things like large proteins and molecules. I'm going to put down here, let me see what I put on the study guide. Yeah, so it does move between 0.2 and 8 millimeters per day. What's interesting though is that when it does move, it moves just as fast axonal transport. It just has long periods of, I guess you can call it, rest. Kind of like two sprinters who are twins. One runs the entire time. The other's going to run, take a break, run, take a break, run, post a selfie on Instagram, run. So it moves, so they both move just as fast, but with slow axonal transport, it seems like there's long pauses, which results in moving less. Okay, and now the question is what about when the axon needs to send things back? I know you're all wondering about that. That is called anterograde. And anterograde transport moves things toward the cell body for recycling. Things like old neurotransmitters, for example, or other cellular debris. So now, how do two neurons interact? Well, recall that a synapse is a connection between either two neurons or a neuron and target tissue. So I'm going to draw a neuron again. Not going to put a whole lot of detail in this one. Just let's get it down. So we call the neuron that is before the synapse, we call it the presynaptic neuron, or before the synapse. Then we have the synapse, and after the synapse, we are going to have the as you can probably guess. You're going to have the postsynaptic neuron. So what's going to happen here is that this space here is called the synapse, and this is the space where the two neurons or the neuron in the target tissue meet. What's going to happen is that neurotransmitters are going to be released from the axon terminal. It's going to be received by either the neuron or the target tissue, and you're going to get a response. Usually, if it's between two neurons, the presynaptic neuron is going to tell the postsynaptic neuron to depolarize. All right, and that is all for the beginning. Next up is going to be glial cells. All right, everyone. Well, we are back, and we're going to cover glial cells. And glial cells are interesting because we used to think that they were only involved with physical support. But in recent years, we found that they're actually involved in many essential operations of the nervous system. And glial cells, glio means glue, and they're far more numerous than the actual neurons are in the nervous system. So these are supporting cells. I'm just going to put down that glial cells are supporting cells. They are supporting, and there are different glial cells found in both the central nervous system and the peripheral nervous system. What I'm going to do here is I'm going to draw a neuron. You'll see why I'm going to draw this neuron in a bit. Keep in mind, I'm just drawing a standard neuron. There are many different varieties and types of neurons. In fact, interneurons in the brain, they have, I believe, some of them have thousands of synapses, thousands of connections. So this is just our basic neuron model. It's kind of like how the cell in the textbook is just a basic cell. It's a template. All right, so this is our neuron. I'm going to draw some organelles just because I'll be bothered if we don't. And can't forget the Golgi. Yeah, all the good stuff. All right, well, the first glial cell we're going to talk about is actually they're basically very similar in function. It's called the oligodendrocyte. And this one is in the central nervous system. And the other one is going to be the Schwann cell. And that one is going to be in the peripheral nervous system, or PNS. So what happens is that how neurons communicate is they are going to receive a signal, usually a neurotransmitter or neurohormone. Usually. So the neurotransmitter is going to stimulate receptors at the site of the dendrite, and then an electrical impulse is going to be generated that is going to go down the axon. Actually, it could be more than a neurotransmitter. It could be some kind of stimuli too, like mechanical stimuli, so on and so forth. So some kind of stimuli is going to activate the cell at the site of the dendrite, or whatever the equivalent is of that. All right, so what this does is that the electrical impulse is going to go down the axon, and then something's going to happen at the axon terminal. Usually there, a neurotransmitter is going to be released. However, the conduction speed is only so fast because the signal has to make its way down the length of the axon. And there are two factors that determine the speed. I know I'm going on a bit of a tangent, but it will all tie in. axon diameter. So the larger the diameter, then the faster the impulse. But the other one is the amount of myelination. Myelination. And the greater the myelination, once again, the faster the impulse. Now, what is myelination? Well, these cells, whether or not you want to, whether or not they're Schwann cells or oligodendrocytes, they are cells that more or less are wrapped around or that are located on the axon. And what they do is that they secrete a substance called myelin. And the myelin is wrapped around the axon, kind of like wrapping duct tape around a pipe, if you want an example of that. So what that does is it actually drastically increases the rate of conduction. That's because under normal circumstances, the impulse would kind of just run down the axon like this. This is how it would happen normally. But when an area is myelinated, instead the impulse jumps. So it's kind of like, what would be faster if you could hop somewhere or walk somewhere? Well, depends how good you are at hopping, but if you're really good at hopping, then definitely hopping. And the spaces between the myelinated regions are called the nodes of Reinvere. Sounds like a name for a band, the nodes of Reinvere. Probably named after the person who discovered it. I think they're also just called now myelin nodes or something like that. Seems to be a movement away from naming things after people. All right, so this myelination is going to drastically increase neuron conduction, and the substance myelin is secreted by the two cells, oligodendrocytes and Schwann cells. Similar function, different location. Okay, now the next one is going to be called an astrocyte. Go ahead and erase this. Astrocyte means star cell. And what this is it is a neuron that has dendritic processes. Kind of looks like a star. So this is a glial cell. It has numerous projections. So this is an astrocyte. And it is found in the central nervous system. Its job is numerous. One of these things is that it stabilizes neurons. So it offers physical support. It does another job, it actually strengthens the blood-brain barrier. I'll just call it the BBB. Not the Better Business Bureau, but the blood-brain barrier. In case you're curious what the about the Better Business Bureau, that was basically Yelp before Yelp. If you had a problem with a business, you reported them to the Better Business Bureau, and they would yell at them for you. And then the company would sometimes say, you know what, you're right, Better Business Bureau. Here's your money back. But yeah, now it's just Yelp, just trash them on Yelp. All right, they also do things like assist with neurotransmitter. Resistance is futile. Assist with neurotransmitter release and uptake. Keep in mind is that astrocytes are still, their true functions are still under investigation, but we do know that they play a lot of roles. Now the next one is going to be something called the microglia. And the microglia, these are found in the central nervous system only. And these are modified macrophages. So this is a microglial cell. And what this does is it is responsible for phagocytizing debris. And what it also does is if there is any kind of infection, it can, which is very rare, it can, in theory, attack pathogens. But the blood-brain barrier, as long as it is intact, is able to resist the intrusion of pathogens. What's interesting is that from research that I've done is microglial cells, they can sometimes be more destructive than helpful. I think I was reading that one of the reasons why neurons in the central nervous system cannot really repair themselves is because the microglial cells are so overactive as far as cleaning up the debris and cleaning up the damage that they prevent the neurons from forming new connections to repair themselves. Neurons in the peripheral nervous system, they do have a limited ability to regrow and repair. The studies I've read a couple of opinions on it, and they're not sure exactly how much of a destructive role microglial cells can play. But I do know that there is some evidence that they can actually prevent neurons from repairing in the central nervous system. There's a condition called meningitis, which is inflammation of the meninges, usually caused by bacteria or virus infiltration. And microglial cells, they send kind of a self-destruct signal to the neurons when they detect the pathogen. And they believe, or some researchers believe, that actually causes many of the neurons in the region to die. It's not the pathogen so much as it is our body's own immune system. So interesting stuff. Next is going to be the ependymal cell. And ependymal cells, they are modified epithelial cells. And they are connected via tight junctions. So ependymal cells. And what these do is that they line certain areas in the brain where there are blood vessels that are present. What they do is that they are an essential part of forming the blood-brain barrier. We'll talk about that more when we get to the central nervous system. And last of all, we have satellite cells. Quickly draw these. Don't want to spend a whole lot of time on glial cells. Satellite cells, they form something called ganglia. Now we're going to talk about this soon, maybe in a couple chapters or so. But one neuron can synapse with other neurons. So this is a neuron. There's the axon terminal. A satellite cell is going to form something called a ganglion, which is a structure where one neuron or a couple neurons can synapse with up to 30 other neurons. I'm just going to draw these kind of sloppily. So what happens is that in this case, all these neurons here, the pregangliotic neuron, because it's before the ganglia, secretes neurotransmitters, and this is going to be received by the postgangliotic neurons, and then it's going to send signals throughout a wide range of the body. So satellite cells form the ganglia, and this region here that I drew is the ganglia. All right, so we covered all of the glial cells. As I said, they are there for support, and they are there for to aid in several essential processes of the central nervous system. Now we're on to events of neuron injury. So I'm going to draw here a neuron. And neurons, they're pretty delicate. And when they're damaged, they usually, as I was saying, they have a poor ability to repair themselves, depending on the location. Neurons in the peripheral nervous system usually have a better job of being able to repair themselves. Some of you, after surgery, you may have experienced, for example, numbness in a certain region of your body, and over time, hopefully that numbness has gone away and you once again had sensation. But neurons in the central nervous system, as I was saying earlier, don't really have that benefit so much. So what's going to happen if there is a neuron injury? Well, the first thing that's going to happen is that we are going to have damage. So let's say that something happens and this area is damaged and possibly even severed. All right, so the first thing that's going to happen is that we're going to have damage, and we are going to have cytoplasm leaks out of neuron. Oh, by the way, this is called neuron injury. Now, recall that all cells, all of the fluid and stuff inside the cell is called the cytoplasm, means basically cell water. So if a neuron is damaged, since it's a cell, it's going to leak out cytoplasm. What's going to happen also is that you are going to have swelling. You are going to have swelling because now what's going to happen is the materials that go from the cell body, the living part, to the axon terminal, there's basically a blockade here. So swelling due to material accumulation. So the materials are basically going to build up at the site of the injury. Think of an axon as a bridge. If just one part of the bridge is damaged, all the traffic is going to be backed up. It's not going to be able to pass the bridge for obvious reasons. Now, recall that cells are constantly communicating with each other. What's going to happen here is the Schwann cells, they are going to send signals to the cell body stating that damage has occurred. So Schwann cells, or if you're in the central nervous system, oligodendrocytes, communicate with cell body. Recall that a cell is not an individual. A cell is like a planet or a country or a city. So one part of the cell needs to communicate with other parts of the cells. Okay. What's going to happen also is that the myelin is going to unravel. And phagocytosis will occur. What's going to happen is that the myelin is going to begin to unravel. And then what's going to happen is that the macrophages, or the glial cells, are going to come and they are going to start to clean up the debris. They're macrophages. That's what they do. That's their job. They're just doing their responsibilities. Now, once this happens, the site that is the distal aspect, as they call it, is going to eventually be degraded, because the proximal side, the side that's connected to the cell body, has an ability to survive. The distal aspect will not. It will eventually be further degraded and phagocytized. Now, if this is the proximal neuron, then we can have regeneration of this distal aspect. If it's in the central nervous system, what usually is going to happen is that the distal aspect will be degraded, but in rare circumstances is that the two sides can bridge. I know there's a lot of research trying to find ways to allow these two ends to be pasted back together almost with molecular glue. So the regeneration part is a question mark. So regeneration depends on severity and location. So just to recap what happens here, neuron is damaged depending on some sort of mechanical stress. You're going to have cytoplasm leakage. And as the materials accumulate, because they cannot pass through the area that's damaged, you're going to have swelling. Schwann cells are going to accumulate with the cell body. Myelin is going to unravel and phagocytosis is going to occur. Depending on the location, this distal aspect will eventually be degraded and phagocytized, or it could possibly be regenerated, or in rare circumstances is that the damage can actually be repaired. But that is certainly not a guarantee. Okay, so we talked a bit about axonal injury. We talked a bit about glial cells. And next up, we're going to talk about, events of an action potential. Sweet. All right, everyone, for the next part of the lecture, we are going to talk about generating an action potential. Now, as we talked about, excuse me, I'm just gonna finish this by putting gates onto these channels. These are going to be channel gates. So as we talked about is how neurons communicate information is they are going to receive a signal of some sort at the dendrites. And then the signal is going to be propagated down the length of the axon using electricity. We call this electrical impulse an action potential, and it resonates down the axon. Then at the axon terminal, you're going to have usually neurotransmitter release. So what you are doing is communicating the signal from the dendrites to the site where the neurotransmitter needs to be released. To show how that happens, we are going to use a cell model. So normally, the cell is negatively charged, or the cell membrane is negatively charged compared to the outside environment. The reason why the cell is negatively charged is because there's lots of stuff inside the cell that has a negative charge. And many cells have a resting baseline. It's called a resting membrane potential of about -70 millivolts. So we're going to talk about the stages down here. So the first stage is going to be called the resting membrane potential. I'll just do MP. It's written out more in the outline. And here are the situations. Well, when the cell is at resting potential, the sodium channels are closed. The potassium channels are closed. And the sodium-potassium pumps are doing their thing. What they are doing is they are going to move potassium into the cell, and they're going to move sodium out of the cell in a ratio of 3:2, 3 sodiums for 2 potassiums. Now, because of the actions of the sodium-potassium pump, you're going to have most of the sodium outside the cell. It's going to draw some sodium. I'm not going to go nuts drawing the sodium. And most of the potassium is going to be inside the cell. And you might be thinking, well, what's the point of this? They both have the same charge. Well, the point is going to be concentration gradients, as we'll see. So both channels are closed, meaning that once sodium and potassium is in its respective environments, it's not going anywhere. You may recall the book talking about potassium leak channels. I'm going to conveniently ignore those just because it's going to complicate an already complicated topic. So let's just get down the get down the foundational knowledge. And the Na plus K plus pumps, one thing to keep in mind, they're always operating. So even though I'm putting down operating, that's only formality because they're always operating, always moving out 3 sodiums for 2 potassiums. Now, whenever a neuron needs to fire or propagate a signal, it needs to generate an electrical impulse along the membrane. Now, you have to ask yourself, how can we make, in this situation, how can we make the cell more positively charged on the inside compared to the outside? Well, let's brainstorm some ideas. Let's say that I decide that I'm going to open up the potassium channels. I'm going to take my eraser here. We're going to open up the potassium channels. And if I do that, you might be thinking, well, that would work. Well, let's think about that. If we take off the gates for the potassium channels, potassium is going to go down its concentration gradient because recall that channels have to be passive. It's going to go down its concentration gradient, and the cell would become more negatively charged compared to the outside. Instead of our curve going like this, which we want, it would go like this, which we don't want. All right, so that's a bad idea. But what if we take off the channels, or we take off the gates off the sodium channels? Well, guess what? We have a very steep concentration gradient of sodium outside and potassium inside. So what's going to happen is the cell is going to go through the second stage, which is called depolarization. It's going to reverse polarity, depolarization. What that means is that the cell is going to have a positively charged electrical environment. The inside of the cell is going to have that positively charged environment compared to the outside. What's going to happen is that sodium channels are going to open, and sodium is going to flow into the cell. Like this. So now we haven't lost any potassium ions, but we've gained sodium ions. Because of that, we are going to have the cell membrane is now going to become positively charged compared to the outside environment. It's going to go up to 0, and then it's going to go up to 30. We call that depolarization. The cell is now going to generate an electrical impulse. As soon as it gets to plus 30 millivolts, we're going to have that electrical impulse being propagated down the axon like this. Once it's generated, it's off to the races. But we don't want it to go above plus 30 millivolts. But we'll get to that in a second. Let's just finish our flow chart. So this time, Na plus channels open. Then we have potassium channels closed still. And the sodium potassium pumps are still operating, but I'm not going to put that down. The cell is going to become positively charged. I do want to note, though, it is very important that the impulse passes what's called a threshold point. So this here is going to be the threshold point, around 0. I'm going to draw it in dots. If the electrical impulse does not exceed this threshold value, it's not going to depolarize. We call this a gosh, why am I blanking on the term? This is, oh, sorry. I was going to say gated. It's graded. It's called a graded potential. I'm going to write that down. A graded potential is a change in voltage without depolarization. So think of it like this. If you touch the desk, you are going to create a graded potential in your nociceptors, your pain receptors, but it's not going to be sufficient stimuli for it to depolarize. Now, if you decide to, in frustration, to punch your desk, don't do that, then you are going to definitely generate enough electrical activity to depolarize those nociceptors. So that's an example of graded potentials. It's like kind of turning it on, but not quite. Like my dryer, for example, if I just touch the button a tiny bit, it'll start to spin, but it won't actually turn on. If I hold the button, though, it will turn on. A graded potential is like when you just tap the button and it just spins for a second and then it stops. So a graded potential, you do not get depolarization. If it exceeds what's called the threshold point, then you will have depolarization, and the threshold point is in yellow. All right, well, the issue is that now we don't want the cell to be positively charged anymore. It's done its job. So now we need to do what's called repolarization. We want the cell to rapidly become negatively charged again. We call this repolarization. Repolarization. So what we're going to do now is we don't want sodium to come in anymore. So what we're going to do is we're going to close the sodium channel. Keep in mind the sodium is still in the cell, but now it's just the channel is going to close, so no more sodium can come in. So the sodium channels are closed. And what's going to happen is now the K plus channels, the potassium channels, are going to open. And the sodium potassium pumps are still going strong. So now we're going to take away the channels of the gates of the potassium channels. What's going to happen is potassium is going to go down its concentration gradient. It's going to exit the cell. So now we're going to have a bunch of potassium go outside the cell. Okay, so we're going to lose some potassium. Now what's going to happen is that this is going to result in something called hyperpolarization. So it's going to go down to, instead of negative 70 millivolts, it's going to go down to about negative 85 millivolts. And that's going to be called, I'll put that here. Let's see, where should I put it here? Hyperpolarization. Cannot write today. Polarization. What this is that the membrane potential becomes more negative than baseline. And the reason why is because, as you can see here, the environment of the cell is in disarray. The ions are in their wrong places. The cell ideally wants potassium inside the cell and sodium outside the cell, so we can have a strong gradient that can result in a strong depolarization. Right now, the ions are kind of mixed and matched. So the reason why hyperpolarization occurs is because right now the cell is not ready to depolarize again. It needs time. It needs time to restore the ions to their correct locations. So the purpose of hyperpolarization, recall that a cell will not depolarize until it passes this threshold value. The purpose is to make it quite a bit harder for that threshold value to be passed by. So it's a protected mechanism that allows the cell to reach resting membrane potential. And guess what? That's exactly what happens next is we have a return to baseline. So now what's going to happen is we are going to go right back to return to baseline, right back to resting membrane potential. So we see here that at resting membrane potential, sodium channels are closed, potassium channels are closed, Na plus, K plus pumps are operating. Let me return the cell to resting membrane potential. Give me one moment here. No, go back. Thank you. All right, so I'll feel better once I get the cell back to where it should be. Okay, great. Let's put some gates on it. So this is how the cell looks at resting membrane potential. When depolarization happens, as we know, the sodium channels are going to open. Sodium is going to begin to go into the cell. It's going to reach positive membrane voltage because now you've added positively charged ions to an environment that already has positively charged ions. Recall that the ions don't really matter. It just is about the charge. Once positive 30 millivolts is reached, the action potential will begin to propagate down the axon like this in a wave pattern. And then we want to get the cell back to its negative membrane potential. So now we're going to close the sodium channels, open the potassium channels, potassium goes out, we return the cell to a negative membrane voltage, and then the sodium potassium pumps will have necessary time to restore the cell to its resting membrane potential status. Now, cells also have channels for chloride. And this basically keeps the neuron off. And that's one thing that I believe benzos do. What they do is that they open up these chloride channels to inhibit the neurons from firing, which creates sedation. I put in here in the outline about gated channels. We've already talked about that. These channels are mainly voltage gated. That's how the sodium channels and the potassium channels know to either open or close, is they're mainly voltage gated, especially the potassium channels. All right, and next up we are going to talk about how neurons actually work and types of neurons. So, see you in a bit. Okay, everyone, well, welcome back. And we're now going to be talking about synapses, neurotransmitters, and types of receptors. So it'll be a very interesting discussion. Now, the first thing we have to do is differentiate between electrical synapses and chemical synapses. There are certain regions in the body where the neurons, they're actually connected by gap junctions, meaning that the electrical impulse runs through this chain of neurons, almost like through a tunnel system. So this is extremely fast communication. As it goes down the neurons, it is going to depolarize a chain of neurons. Now, this is good for tissues where contraction must happen in a sequence or all together. For example, cardiac muscle. The pacemaker cells, when they propagate the action potential, it races through a number of the cardiac muscle cells, which helps them to depolarize all in the sequence. So that's one place where you would find electrical synapses. You don't see any chemical signals passed down. It is just gap junctions combining neurons and allowing for them to all depolarize as a unit. Now, as far as chemical synapses, these are traditional forms of neurotransmission. And the first one we're going to observe is something called, well, a neurotransmitter. So what I'm going to do is I'm going to draw what's called a presynaptic neuron, like so. So this is the axon terminal I'm drawing here. I know it looks like a nose. I'm sorry. Okay. Maybe tomorrow I'll draw one that looks more like a cone. For whatever reason, I have a hard time drawing consistent axon terminals, but that is a me problem. Now we see here that in neurotransmission, let me label this on top, we see that there is one axon terminal and one neuron dendrite. So it's neuron to neuron. So we see here, we see a synapse. So it is from neuron to neuron, which I know you're probably saying, well, yeah, that's what neurotransmitters do. They go from neuron to neuron in the classical sense. Now, neurotransmitters usually use a receptor ligand complex called an ionotropic receptor. So I'm going to draw that here. And neurotransmission generally uses what's called an ionotropic receptor. These are very simple. They are extremely rapid. They are extremely fast acting. And that's because ionotropic receptors involve ligand channels, meaning that ligand binds with the receptor and it opens a channel. One example of that is going to be the neurotransmitter called acetylcholine, which we'll talk about that one in a bit. And the receptor is called a nicotinic receptor. So that means acetylcholine binds with the nicotinic receptor. And when that happens, it's going to open up a sodium channel. And a plus channel opens. And then sodium is going to rush into the neuron, into the postsynaptic neuron, and you're going to have depolarization. Neurotransmission is when two neurons synapse join at a synapse. The presynaptic neuron is going to release neurotransmitter. The neurotransmitter binds with the receptor, and it usually is going to open up a channel. So if it is called a neurotransmitter, it is going to follow these guidelines. One neuron to the other, and then an ionotropic receptor. However, we have another term that you probably haven't heard too much of. It is called a neuromodulator. Let me go ahead and clean up this mess here. Neuromodulator. We haven't talked about this yet. In fact, we're not going to talk about it for a couple chapters. But there are different types of neurotransmission. Sometimes it is not from neuron to neuron. Sometimes it's from neurons to groups of neurons. In fact, I'm going to draw you an example of a modified neuron called a varicosity. So instead of the axon terminal looking like this, let's draw this really fast so you can see what I'm doing here. Excuse me. Now, so far we are used to seeing the axon, then we're used to seeing the terminal axon, which is this thing here. Well, with certain modified neurons, we don't see the classical axon terminal. Instead, we see something that looks like this. We see that there's actually diffuse regions of these, like almost things that look like bubbles. And I'll show you why that's important. We'll be talking about this more in a couple chapters. So these are the varicosities. So what happens is that these varicosities they're actually going to sit above a muscle tissue of some sort, a muscle tissue or a group of neurons. And the varicosity is going to look like this. And I'll just say that this is going to be cardiac muscle tissue on the bottom. Cardiac muscle tissue is going to have receptors for this. And what's going to happen is that the chemical messenger is going to be contained in vesicles. The vesicles are going to fuse with the membrane, and they're going to rain down all throughout the tissue. So it isn't neuron to neuron, it's neuron to group of neurons, or in this case, neuron to tissue. So it is called a neuromodulator when several neurons or surface area is involved. And by surface area, I just mean just a wide area on top of a tissue. And it also needs to be metabotropic. Now you're probably saying, all right, there's Professor with his big vocab again. Metabotropic receptors are GPCRs. So a metabotropic receptor, actually let me write it up here where we have a little bit more room. Metabotropic receptors. Come on, I can fit it. There we go. They are GPCRs, meaning that they have second messenger pathways, and they're also a little bit more slow acting. By slow acting, maybe it takes a couple milliseconds to work instead of microseconds. Still relatively fast compared to our standards, but slow compared to ionotropic receptors. So when something meets this criteria, is the receptors are GPCRs, metabotropic, and it involves several neurons or a wide surface area, then we call it a neuromodulator. Serotonin, for example, is a neuromodulator because it involves wide areas or several different types of neurons. Dopamine is a neuromodulator. In fact, I believe, and I'll have to double check this, dopamine, even though in the brain it's regarded with feelings of reward, I believe it is a beta-1 agonist on cardiac tissue, meaning that it accelerates heart rate. I'll have to double check that though. I tend to have a bad habit of speaking off the cuff without really double checking things, but as they say, a broken clock is right twice a day. All right, so now we're going to talk about types of neurotransmitters and neuromodulators. One that we're going to be talking about a lot, and this is one of the most ubiquitous in the body, is what we call a small molecule neurotransmitter. And the one that we're going to talk about a lot is going to be acetylcholine, which is actually made up of two small molecules, acetyl-CoA, which if you remember our friend, the citric acid cycle, cellular respiration, I'm sure many of you look upon that with fond memories. And also another small molecule called choline. You combine them and you get acetylcholine. And the reason why acetylcholine is so important is because one complex is going to be acetylcholine plus a receptor called nicotinic receptors. And nicotinic receptors, we'll be talking about this a lot, so if you don't catch it the first time, that's okay. They are ionotropic. And what they do is that they depolarize adjacent tissues, especially muscle tissue. This is the chief neurotransmitter responsible for muscle contraction. Acetylcholine, when it binds with a nicotinic receptor, it's going to open up sodium channels on that muscle fiber, which is going to depolarize the muscle fiber and result in contraction. All three types of muscle are triggered by acetylcholine. However, acetylcholine also can bind with a metabotropic receptor. It's called muscarinic, which, by the way, what they have done in science is they found agonists usually through plants, through plant hormones. So what sorry, I'm not focusing on what I'm saying. So what they've done, for example, is they took a compound from tobacco called nicotine, applied it to cells, and they could actually see where those cells were, which receptors they bound to. So they go, oh, okay, well, nicotine binds with this receptor here, so we're going to name it after the agonist. We're going to name it nicotinic receptors. It's the same thing with here. Is they found a mushroom toxin called muscarine, saw where that bound, and they called it a muscarinic receptor. Now, this is involved with quite a few parasympathetic processes. It is metabotropic, meaning that depending on the type of receptor isoform it binds to, the effects might be slightly different. And it's also involved with many parasympathetic responses. For example, the neuromodulator norepinephrine is going to bind to cardiac tissue and is going to increase heart rate. However, if acetylcholine binds with muscarinic receptors on the heart, it's going to decrease heart rate. Okay, now this and we'll talk about this a bit later too, but when acetylcholine is released into the synapse, it's also rapidly broken down by an enzyme called, let me vary up my pen colors a bit, acetylcholine esterase. I think I spelled that right. And this is a catabolic enzyme that degrades acetylcholine into acetyl-CoA plus choline. You don't want acetylcholine to stay in the synapse any longer than it has to, because the muscles will become overstimulated. We'll talk about that later on. Now we have what's called the amines. We talked about this a bit, but examples of amines are going to be synthesized from two types of amino acids, and recall they don't need to be made via transcription translation, because these are single amino acids. We have tryptophan, and we also have tyrosine. So these are two amino acids that are modified to form neurotransmitters or neuromodulators. Tryptophan, we have serotonin, We have dopamine. And we know that they're amines because they literally end with amine. And histamine. And tyrosine is involved with catecholamines, epinephrine, and norepinephrine. And as I said before, in the UK, these are called adrenaline and noradrenaline. So epinephrine and norepinephrine fall under the wider category of catecholamines. Now, speaking about these, is examples, as I said, are going to be called catecholamines. These are examples of amines. Give you a preview of when we talk about the autonomic nervous system. But catecholamines, they bind to a class of receptor called adrenergic receptors. Now, both epinephrine and norepinephrine can bind with each of the adrenergic receptors. However, certain adrenergic receptors have a certain affinity toward epinephrine or norepinephrine. So either epinephrine or norepinephrine can bind with the adrenergic receptors, but the affinity is different, meaning that if epinephrine is present, then it's more likely to bind with, I don't know, for example, an alpha-2 receptor than norepinephrine is. So let me clarify this a bit. I just don't like the way this is written. So catecholamines. We have epinephrine, as I said. And we have norepinephrine. And these bind with a class of receptor called adrenergic receptors. There's 4 isoforms that we talk about. There's a 5th one too, but one of the ones we're going to talk about is one that's called alpha 1, which is responsible for negative feedback. Of norepi release. So keep in mind is that both epinephrine and norepinephrine, they both bind to the same class of receptors. We have alpha 2, which is found on smooth muscle and causes contraction, meaning it's going to make, so it's found a lot on blood vessels, and when the catecholamines bind with alpha 2 receptors on the blood vessels, it tells them to become more narrow or to constrict, which restricts blood flow in that area. Why that is, you'll learn in a couple chapters. Beta 1, these are found on cardiac tissue and causes increased what we call cardiac output, which is making the heart beat more often. So increase heart rate and increase the strength of contraction. And we also have beta 2, which is found on bronchioles. That's not how you spell bronchial. Bronchioles. And skeletal muscle. And it causes dilation, meaning that It's going to open up the airways, or it's going to open up the blood vessels and skeletal muscle, so more energy and nutrients can go to your muscles. Because of course, catecholamines are designed to cause what's called a sympathetic response. So we're seeing that we have the same neuromodulator. However, its actions are different depending on which adrenergic receptor it binds to. And in case you were wondering, adrenergic receptors are examples of metabotropic receptors because they are G protein coupled. Now we are going to get into larger neurotransmitters. These are called examples of this, and I'm not going to write this down because it's pretty good on the outline. But glutamate is a very, actually, I'll write it down. I'm just being lazy. So we have now amino acids, and these are made in the cell via transcription translation. And one of those examples is glutamate. I believe that glutamate might be the most prevalent amino acid in the body, and that there's actually several different types of receptors. One is called AMPA, which we'll talk about, and AMPA is a ligand-gated channel, so it is an ionotropic receptor. And the other isoform is going to be called NMDA. NMDA. And this is actually going to be a, it's also going to be the ionotropic, but it's going to be very complex. We'll be talking about these later on. And it also binds to something called a GABA channel. And GABA channels, when they are activated, they're going to be inhibitory. So these are also going to be ionotropic. And what it does is it is going to cause inhibition. So it's going to tell neurons to turn off. There are other things that can act as neurotransmitters: peptides, gases. For example, there's a gas called nitric oxide, which also acts as a neurotransmitter of sort. So those are examples of neurotransmitters. All right, so next what we are going to do is talk about, the steps of synaptic signaling. That'll be a good time. So I'll be back in a bit with another lecture. All right, well, we are now on to the steps of basic neurotransmission, using acetylcholine and nicotinic receptors as a template. So how does neurotransmission occur? What is the stimulus that is going to allow the release of the neurotransmitter into the synapse? And then what happens when the neurotransmitter binds with receptors? Well, we're just going to use this as a template because we need some understanding of how neurotransmission occurs. Just to tell you, this here is called the postsynaptic tissue, which is muscle. And this here is called the presynaptic neuron. So let's go ahead and define some of our terms here before we get started. So these receptors sitting on top of the muscle tissue are called nicotinic receptors. If you recall, they are ionotropic and connected to a channel. So I'm just going to go ahead, this is not in the legend, I'll go ahead and add it in, sodium channel. So they're ionotropic. As soon as acetylcholine binds with nicotinic receptors, channel opens. Now, in this blue bubble, we have acetylcholine, abbreviated ACH. In the vesicle, these green things here are called snare proteins. These channels colored in black, they are voltage-gated calcium channels, and the black dots are calcium. And I believe, oh, and this square here in orange is going to be called acetylcholinesterase, and that is the enzyme that degrades acetylcholine. So green things, snare proteins; sky blue things, acetylcholine. These channels in black are going to be voltage-gated calcium channels. The black dots are going to be calcium. The pink receptor is going to be nicotinic receptors. And this brown channel is going to be a sodium channel. And I'm not going to draw sodium here. You'll get the idea. Now, the first thing that is going to happen, so I'm going to do the steps up here. And the steps are also on your outline, but I suggest you write them down because it is just a good habit. So, steps. Well, the first step is going to be the neuron is going to depolarize. So if you recall, an action potential has been generated. Oh, I would be remiss if I did not draw some mitochondria. Sorry, gotta do that. There's mitochondria in the axon terminals, and they provide energy to synthesize neurotransmitters. So we'll do the mitochondria in this nice turquoise. Okay, but anyways, the neuron is going to depolarize. The neuron is going to generate an action potential when it is stimulated, and the action potential is going to end at the axon terminal. So we'll go ahead and just choose normal blue for that. So we're going to have an action potential. Oh, it's abbreviated AP. Now when that happens, voltage-gated, which I'll abbreviate VG, voltage-gated Ca2 plus channels open. So that's basically the entire purpose of depolarization, to open up these voltage-gated calcium channels. Now when that happens, we are going to see the gates open, and there's lots of calcium outside the cells. That's also why we have so many mechanisms to break down bone into free blood calcium, because we need calcium to do things like diffuse into the neuron passively. And calcium is going to bind to these snare proteins and activate them. When these snare proteins are activated, they're going to latch on to the vesicles, and they're going to help them fuse with the membrane, like so. When they fuse with the membrane, we are going to have calcium-mediated exocytosis, meaning that the calcium is going to assist in the neurotransmitter being released. So that's going to be step 2. So Ca2 plus diffusion, and then calcium-mediated exocytosis. Okay. So what's going to happen is that the law of diffusion is going to occur, because you have all of these neurotransmitters all bunched up, all bundled up. They don't want to be bunched up and bundled up, so they are going to diffuse from the presynaptic neuron to the postsynaptic tissue. So what's going to happen here is that the neurotransmitter, in step 3 as I'll put down, is going to diffuse and bind with these nicotinic receptors. So we're going to do step 3. Neurotransmitters diffuse and bind with receptor. So now we need to terminate the process. As we'll talk about later when we talk about tetanus, we really do not want acetylcholine to stay in the synapse for too long. Actually, it looks like I skipped a step here. But anyways, whether or not you want to write it down like I did or refer to the outline is the same information. I have four steps on the outline for the actual process of neurotransmission, but I only put down 3. Same information though. So now we're going to do steps of termination. Starts with 5 on your outline. Because now we need the process to stop. We don't want it to happen anymore. So steps of termination. So there's basically 3 ways that this process can stop. Step 1 is going to be reuptake. You probably have heard of a process called, or a class of drugs called serotonin or selective serotonin reuptake inhibitors. And that's because presynaptic neurons, they have active channels, they have pumps, they are called reuptake pumps. Let me find a good color for this. Yellow, sure, why not? And these are actually pumps that are on the presynaptic neuron. And what they do is that they basically take back up the neurotransmitter. We call this process reuptake. And you're probably saying, okay, well, what's the benefit to that? Well, recycling. Because if you can take back something, you can reuse it again. The next way is going to be passive diffusion. I don't know if I put this on here. There's actually 3 ways. I put on the outline, there's only 2. We're just going to go ahead and say that there's going to be 3, because the other one is going to be passive diffusion. So what happens here is that the excess amounts of neurotransmitter, they just go away. They just diffuse out of the synapse. So they diffuse out of synapse. So they just leave. It's kind of like you were to stand on top of a storm drain and pour a bag of M&Ms on the ground. Yeah, you'll see lots of them will just go in the storm drain and disappear. It's kind of like what's happening with this, except there's thousands of Skittles in this example. Last one is going to be a process called hydrolysis, or as I like to call it, hydrolysis. Hydrolysis. What that means is that you break down a complex molecule into 2 simpler ones using water. And what that's going to be is that's going to be through an enzyme-mediated process, acetylcholinesterase. So what this enzyme is going to do is it's going to very rapidly, by the way, there's several of these enzymes here, is they are going to break down acetylcholine into acetyl-CoA and choline. So this is done through acetylcholinesterase, and it's going to take acetylcholine, going to convert it into acetyl-CoA plus choline. Now, even though this might seem like it's happening very slowly, it actually happens very fast. We're talking microseconds. So this entire process that I just went through has happened untold thousands of times since I even brought it up. So anyways, the steps of it, just to recap, is the presynaptic neuron is going to depolarize. what's going to happen is that voltage-gated calcium channels are going to open. When they open, calcium is going to diffuse in, and it's going to activate the snare proteins, and the snare proteins are going to trigger exocytosis of the neurotransmitter containing vesicles. This will release the neurotransmitter into the cleft. It's going to rain down via passive diffusion to the receptors, and it's going to depolarize the muscle tissue. As soon as this happens, we need to end the process. One way to do that is through reuptake, where there's pumps on the presynaptic neuron that take back the acetylcholine. The other is passive diffusion. It just leaves. It just goes away. And the last one is hydrolysis. You have an enzyme, acetylcholinesterase. What this does is it catabolizes, breaks down acetylcholine into acetyl-CoA plus choline, breaking it down and thus giving it materials to make more neurotransmitter. All right, well, I hope you all enjoyed this talk. We are done with chapter 8. So if you have any questions, you know where to stop by. All right, over and out, everyone.